Welcome, everyone. We're going to wait just a minute or so to let everyone into the Zoom. Okay, let's get started. Well, welcome, everyone. This is Christy Sullivan. I'm the Secretary of the American Society for Cellular and Computational Toxicology. What we have for you today is something a little bit different. It's a special virtual session, which we're presenting today as part of our 14th annual meeting, which took place in October of 2025 in Gaithersburg, Maryland, outside of Washington, D.C. If you'll recall, we had some people were unable to attend the conference or speak or present because of the government shutdown and some other issues related to that. And so we are very lucky that we were able to get the speakers to come and present their work in this special virtual session. Before we get started, a few announcements. I encourage you to visit our websites to find out more about our organization, ASCCT, and please consider becoming a member to support programs like this webinar series, all of our other webinar series, and of course, the student awards and travel awards and annual meeting and everything else that we do. The webinar is recorded, and we will have the recording posted on the ASCCT website in a few days. You can ask questions in the webinar platform using the Q&A feature at the toolbar at the bottom of your screen. Please do not use the chat to send us questions. It's too hard to keep track of questions in two places. So look for that little Q&A button and send us questions at any time during the webinar. There's going to be a short Q&A period after each presentation is finished, and then another Q&A period at the end. We have also enabled closed captions, and you can turn them on or off as you like. And please say hello and let us know where you're joining from in the chat. Before we get started, just to let you know, we are going to have a 15th annual meeting this year. That is going to be in Research Triangle Park, North Carolina. Likely, we have our tentative dates, October 28th through the 30th, and with a Society for Adverse Outcome Pathway ancillary meeting on the day previous. We are hoping to confirm the venue and the dates very soon. And we will also open up session and CE proposals next week, and those will be due on May 1st, with regular submissions and travel awards deadlines coming after that. We also have a very full calendar of virtual webinars, both presented by ASCCT and some of our sponsoring organizations. So as always, feel free to take a look at our calendar and register for those events. So today, as I mentioned, what we have is a full meeting session titled Advancing Cannabis Safety Assessments with New Approach Methodologies. We're featuring 5 U.S. Food and Drug Administration researchers. The session was put together by Yitong Liu and features Michael Santillo, Xiugong Gao, Kirsten Eckstrum, and Piper Hunt. I'm going to introduce everybody with a short introduction first, and then we'll get started with all the talks. You could find their full bios on our website if you want to learn more about their backgrounds. And before I introduce everyone, I just want to say thank you so much to the chair and the presenters for pulling this together. All right, so our speakers first is Dr. Yitong Liu. She is a senior research pharmacologist and diplomate of the ABT with over 15 years of experience in regulatory science at the FDA's Human Foods Program. She specializes in using new approach methods, including quantitative structure-activity relationship modeling, physiologically based pharmacokinetic modeling, and in vitro assays to evaluate the safety and pharmacokinetic properties of food substances, particularly dietary supplements and phytochemicals. Dr. Michael Santillo is a senior research chemist in the FDA's Human Foods Program with over 15 years of experience specializing in toxicology, and his research centers on developing, applying, and combining multiple new approach methods to evaluate the safety of chemicals in foods and dietary supplements. Dr. Xiugong is a research biologist in the Office of Chemistry and Toxicology, Human Foods Program at the US FDA. He serves as principal investigator for the toxicogenomics and stem cell research programs and has more than 20 years' experience applying transcriptomics and human in vitro models to toxicity testing and chemical safety assessment. Dr. Kirsten Eckstrum is a research biologist in the Division of Toxicology. Her lab work focuses on evaluation of new approach methods that can potentially be utilized to advance food safety evaluations. And she focuses on a tiered approach for the evaluation of potential hepatotoxicity of botanical extracts, utilizing both rapid and microphysiological systems. And finally, Dr. Piper Hunt is a research biologist at the FDA Human Foods Program, where she's a leader in the development of novel alternative methods for toxicity assessment. She has over 2 decades of experience spanning molecular genetics, nutraceutical assessment, toxicology, and alternative model development. So with that, let me turn it over to our first speaker, Dr. Liu.
Can you see my screen now?
Yes, and we can hear you.
Okay. First, I would like to thank the meeting organizers, especially Christy and Allison, for being so flexible and then supportive to put this session together. And it's long overdue. And then I'm excited here to share our work with the ASCCT community. So I'm the first speaker. I'm going to give you an overview of the cannabis toxicity and human use. And then following speakers will talk about using specific names to investigate cannabis and then cannabinoid safety. So in my presentation, I'm going to first talk about the history of cannabis use and the regulatory landscape. Then I'll introduce the cannabinoids. Following that, I'll talk about adverse health effects of cannabis and the cannabinoids. Lastly, I'll talk about consumer use of cannabis products. Cannabis has been cultivated for more than 5,000 years, beginning in Central Asia and then spreading globally through trade routes. In the US, cannabis was widely grown and used medicinally in early America, but was federally banned in 1937 by the Marijuana Tax Act, and later classified as a Schedule I substance under the 1970 Controlled Substances Act. It then returned with state legalization for medical use in 1996 in California, and then recreational use in 2012 in Colorado and Washington. If you look at this picture on the right, cannabis has 2 main strains, the marijuana strain and the hemp strain. The marijuana strain has 5 to 25% of THC content, while the hemp strain has no more than 0.3% of THC. This is the major difference between the 2 strains. The Agriculture Improvement Act, which is also called the Farm Bill of 2018, removed hemp from regulation by the Drug Enforcement Administration under Schedule I of the Controlled Substances Act. The 2018 Farm Bill defined hemp as cannabis sativa L with delta-9 THC concentration no more than 2.3% on a dry weight basis. In November 2025, Congress adopted amendments tightening hemp THC rules. For example, the new rule used the total THC to replace the delta-9 THC in the definition of hemp. It also set a cap for the total THC content per finished product. Additionally, it includes the synthetic cannabinoids. Hemp products remain subject to FDA regulations under the Federal Food, Drug, and Cosmetic Act, including drugs, foods, dietary supplements, cosmetics, and veterinary products. Here's a list of the currently FDA-approved cannabis products. They're all drugs. Marinol is a synthetic delta-9 THC, which was first approved in 1985. It is used to treat nausea from chemotherapy and AIDS, anorexia. It's currently under Schedule 3 under the CSA. Sesame is a synthetic cannabinoid, first approved in 1985 and reapproved in 2006. It's used to treat nausea from chemotherapy. It's under Schedule 2. Santillo is a synthetic delta-9 THC, which was approved in 2016. It's used to treat nausea from chemotherapy and AIDS, anorexia. It's under CSA Schedule 2 as well. And lastly, most recently, Epidiolex, which is a CBD product. It was approved in 2018. It's used to treat childhood seizures and tuberous sclerosis complex. It used to be under Schedule 5, and then it has been removed from schedule. In addition to the approved cannabis products, there are some unapproved cannabis products on the market. Some of these products are potentially dangerous to consumers, especially the copycat food products containing delta-8 THC and the dietary supplements containing CBD. In 2023, FDA has concluded that the existing regulatory frameworks for foods and supplements are not appropriate for cannabinoid, for CBD especially. And then FDA will work with Congress to find a new authority to regulate these products. This is still in the works. So now I'm going to introduce cannabinoids. Cannabis contains large numbers of secondary metabolites, including cannabinoids. These compounds accumulate primarily in the female flowers and leaves. There are two major cannabinoids, delta-9 THC and CBD. Additionally, there are more than 120 other minor cannabinoids. Some of these cannabinoids are called cannabis-derived cannabinoids. They occur naturally in the plant. They're extracted directly from the plant. Examples include delta-9 THC, CBD, CBG, CBN, and CBC. While some other cannabinoids, they're called cannabis-related cannabinoids. They're synthesized in the laboratory. Some synthetic cannabinoids may also occur naturally in the plant, and some may not. For example, delta-8 THC is synthetically derived, and it also occurs naturally in the plant. While navalone is only synthetic, it doesn't exist in the plant. There are at least 11 cannabinoid types: the THC type, CBD type, CBG type, and at least more than others. Within each type of these cannabinoids, there are multiple compounds. They normally share the same core structure and have some variations on the side chains and the ring. For example, these are the THC type cannabinoids. Upper left, you see delta-9 THC. On the right, you see delta-8 THC. The only difference between the two compounds are the position of the double bond. And then lower left, you see delta-9 THCV. Compared to the delta-9 THC, the section on the delta-9 THCV is shorter. In the middle, you see delta-9 THCA, which has a carboxylic acid on the ring. So the last compound is HHC. So on the ring, it has a saturated ring instead of a double bond compared to the delta-9 THC. Next, I'm going to switch topic and talk about the adverse health effects of cannabis and the cannabinoids. In cannabis, marijuana has high delta-9 THC content, so it's intoxicating. While hemp, which has a low delta-9 THC content, is minimally intoxicating. In cannabinoids, the THC-type cannabinoids may cause intoxicating effects, while CBD is not intoxicating, but can cause adverse health effects at high doses. The majority of the minor cannabinoids, the safety is largely unknown. On the right, I give you some examples of the intoxicating cannabinoids and the non-intoxicating cannabinoids. So now I'm going to talk about the adverse health effects of the selected cannabinoids. First one is delta-9 THC. It can cause negative cognitive and neurological effects, such as impaired memory and learning, distorted perception, loss of coordination, drowsiness, and anxiety. In the cardiovascular system, it may cause increased heart rate and elevated blood pressure. In the respiratory tract, it can cause throat and lung irritation, coughing. Long-term regular use can lead to physical dependence and withdrawal following discontinuation, as well as psychological addiction and dependence. For CBD, in the GI tract, it may cause diarrhea and decreased appetite. In the neurological system, it may cause drowsiness or sleepiness. In the liver, it may cause elevated liver enzymes and then interact with drugs metabolized by certain liver metabolizing enzymes. In animal studies, CBD may cause damage to fertility in males or male offspring of females who have been exposed to CBD. The next is delta-8 THC. This is the isomer of the delta-9 THC, and then it's also psychoactive and intoxicating. Between 2020 and 2022, there have been 104 active reports of adverse events related to delta-8 THC, such as hallucinations, vomiting, tremor, anxiety, dizziness, confusion, and loss of consciousness. There are more than 2,300 poison control center case reports related to delta-8 THC, including one pediatric death. FDA strongly advise against the use of CBD, THC, and marijuana in any form during pregnancy or while breastfeeding. It may cause higher risk of a premature birth and stillbirth, impair the brain development in fetuses and newborns, and potential reproductive effects in developing male fetuses. Then next, I would like to talk about the consumer use of cannabis products. There are many cannabis products on the market, including tinctures, capsules, topicals, built-in personal care products, vape oil and cartridges, combustible and flowers, pet products, gummies, beverages, and other edibles, as well as a food drug. For CBD, the market has surged after the 2018 Farm Bill, but has since declined. The 2025 forecast by the Redfield Group is a $2.8 billion total sale of CBD, including around $2 billion for non-pharmaceutical products. The top 3 reasons for consumer use of CBD is for physical relief, sleep, and relaxation. It's important to point out that it's currently illegal to market CBD by adding it to a food or labeling it as a dietary supplement. For delta-8 THC, in the second quarter of 2024, 5.6% of U.S. adults surveyed by the Redfield Group reported delta-8 THC use. How do people use it? People use it as edibles such as gummies and brownies, tinctures, vaping and smoking, capsules, creams, lotions, or patches. Delta-8 THC is often referred to the marijuana light or diet weed because it provides a milder intoxicating effect. So some of these delta 8 THC products are pretty dangerous because they mimic chips, candies, and snacks from popular national brands. These products pose a special risk to the tumor. If you recall earlier, I mentioned like in November 2025, Congress has tightened the definition of hemp to include a cap on the total THC content. This law has a delayed effective date, which will be the November of this year. So after the law takes effect, that will effectively ban all the delta 8 THC on the market. Key takeaways. Cannabis-derived products contain a wide range of cannabinoids, both occurring naturally or derived synthetically, many of which have a limited safety data. Cannabis use can cause serious adverse health effects, especially in vulnerable populations. More research is needed on the safety, chemical composition, and potential interactions with other consumer products. In 2024, FDA released a roadmap to reduce animal testing in preclinical safety studies. This roadmap outlines a strategic, stepwise approach for FDA to reduce animal testing and to include NAMS studies in safety assessment. In the following presentations, you will hear FDA's research on the safety of hemp and the cannabinoids using various in silico and in vitro NAMS. Thank you. And then that'll be my presentation. I'll stop sharing.
Yeah, Dr. Liu, we do have one question.
Okay.
Is there any difference between synthetic and natural delta-9 THC in terms of their effects or toxicity, other than difference in purity?
They're all cannabinoids. They have some of the cannabinoids are intoxicating. It could be both naturally occurring or synthetic. Like delta-9 THC is naturally occurring, but delta-8 is mainly synthetic, although it occurs really small amount in the plant as well. So it's hard to say in general, like the naturally occurring is intoxicating, but synthetic or not. They can have similar or different effects and toxicity.
Okay, thank you. Okay, our next presenter is Dr. Michael Santillo. Michael, we can see your slides. Everything looks good.
Okay.
Go ahead.
All right. Thanks so much. Thanks to Yitong Liu for putting together this seminar session, and also thanks to ASCCT for hosting. So today I'm going to talk a little bit about some broad screening of cannabinoids on various safety-related biological targets in an effort to understand some of their potential health effects. So this is probably basic review for everybody, but I just want to talk briefly about adverse outcome pathways. So it's a concept in order to kind of explain the steps from the initial steps of molecular initiation to final adverse outcomes. So in an adverse outcome pathway, we have our molecular initiating events. So this is where chemicals interact with different biomolecules. In the context of this talk, I'll be talking mostly about different protein targets like enzymes, transporters, ion channels, and receptors. So these events lead to downstream effects called key events. These are changes that occur in the cellular, tissue, or organ system function. And then finally, we have our adverse outcome or observed toxicity. So in this talk today, I'll be talking about how we can predict the binding between the cannabinoids and targets, and also measure them using in vitro methods as well. So if you think about it, there's probably tens of thousands of proteins in the body. And some of these are more well known than others in terms of their effects on safety. So about maybe 15 years ago, a bunch of pharmaceutical companies came together and established this panel of biological targets. and there's around 44 targets on this panel. And the reason why they chose these targets is because they have very well-known and historical data supporting their links between these targets and adverse health effects that they observe. And the pharmaceutical companies often screen their candidates using these assays and then include them in INDs to the FDA. And the FDA actually looks at that data in addition to animal data before initiating the first in human studies. So in this project here for the cannabinoids, we use this panel of targets, and you can see that a majority of these targets are G protein coupled receptors, followed by ion channels, enzymes, membrane transporters, and a few nuclear receptors as well. And here's some examples, or not some examples. These are the ones that are actually in the panel. So you can see that in general, if you're familiar with these targets, a lot of them are related mostly to the nervous system and cardiovascular system. Although, the effects on these targets can also affect other organ systems as well through the nervous system. But those are the main focus of these targets. So just to briefly talk about the methods, this is just an initial screening project. So we mostly perform competitive ligand binding assays. So with competitive ligand binding assays, here you have your target. In this case, it's a receptor. It's in solution. It's a membrane preparation. So you grow you culture cells containing the receptors, and then you lyse them. So they're basically homogenate. And so you take this solution and you incubate it with a very specific ligand that's radiolabeled. And what happens is after you incubate and wash, you have your specific ligand that's still bound. And this basically results in no inhibition of the targets. And this represents a negative control. Now, if you add a test compound and it binds to the target, it'll compete for binding to the receptor. It'll displace the radiolabeled ligand. So after washing, you end up getting a reduced signal. And in this example here, we could see the test compound's a very potent inhibitor. And we simply record the inhibition over a log of concentration range and obtain concentration response curves. And after that, we can get our IC50s, which represent the potency of that binding interaction. So the first target I want to talk about is the cannabinoid receptor. Obviously, these are cannabinoids, so this is the one of most interest to everybody. In particular, I'm going to talk about cannabinoid 1 receptor. So cannabinoid 1 receptor is found throughout the body, especially the nervous system. And this is a list of different adverse health effects that are associated with this receptor. These are pulled from a database called OffX. I don't have time to go into details on that database, but later on I can explain more if you're interested. So basically, this cannabinoid 1 receptor, compounds or drugs that we know bind to it cause a variety of adverse health effects that are very dose dependent. So you can see what I did here is I took the effects and I categorize them by organ system. So you can see in this first group, we have a lot of cardiovascular effects. Second group is gastrointestinal. We have the lung, and finally the nervous system. So the cannabinoid receptor is definitely a very important target when trying to understand the safety of chemicals, especially cannabinoids. So first, if we talk about non-THC type cannabinoids, in general, these are very weak agonists on the cannabinoid receptor. And because of that, they don't cause any intoxicating effects. That doesn't mean they can't cause any effects in the nervous system, but they don't particularly cause intoxicating effects. So here are some examples of non-THC type cannabinoids. We have CBD, the binding concentrations around 4 micromolar, CBDV, and also CBG. And these are all fairly weak in the micromolar range. And another reason why that we don't see any intoxicating effects is because typically the internal concentrations in the body are maybe like 100 or 1,000 or even more times less than these concentrations here. So you wouldn't really expect any activity through the cannabinoid receptor. Now, if we talk a little bit more about the THC type, and you know, in Yitong's introductory talk and presentation, she talked a lot about these THC compounds, and I'm going to talk more about their cannabinoid receptor binding. So here we have delta 9 THC, and this binds to the cannabinoid receptor around 30 nanomolar. Here we have delta 8 THC. It's fairly similar, in the similar range, around 30 nanomolar as well. So by changing the double bond, we can see maybe some changes, not too much. And then if we take this ring here and we turn it into an aromatic ring for CBN, we can see we get about a you know, tenfold loss in potency. And then finally here we have HHC, which is hexahydrocannabinol. So now we're saturating this ring, and we also have a loss of potency in between the delta 9 and the CBN. So it's around 150 nanomolar. So you can see that how the structure of this ring plays a really important role in potential cannabinoid receptor binding. Another thing we can do is we can also change the length of this carbon chain. So in general, as we increase this chain length, we see a very increased potency. So this example here shows delta 9 THCP. So we're adding 2 methyl groups here, and we can see that the potency goes down to 1 nanomolar. And then finally, another change we can make to the THC molecule is by adding an acid group. And by adding an acid group here, we see the greatest loss in potency among all these. So we see about 800 nanomolar binding constant. So you can see the acid group almost takes away all of the binding. So that's an overview of the CB1 receptor. So now I'm going to talk more about some of the studies we did on some of these other targets as well, in addition to the CB1 receptor. So these are the list of cannabinoids we studied when we screened it against this 44 target panel of targets. So you can see we have a very wide variety of cannabinoids here. We have CBG, which contains 1 ring. CBN has 2 rings. We also have acids, so we have CBGA and CBCA and CBDA. So we selected these cannabinoids mainly because of their popularity. We wanted to test some of those THC cannabinoids as well, but they're really difficult to work with because they're controlled substances, and you can't just, you know, buy them, put them in DMSO and send them off to labs for testing. So we were limited to basically these cannabinoids here. So first, we just screened them at 10 micromolar on the panel of 44 targets. So you can see a heat map of the cannabinoids on the left, the various targets as columns, and the binding is represented on a blue color scale. So when you look at this initial testing at 10 micromolar, you can see the cannabinoids were very different in terms of their binding profiles. CBG was fairly promiscuous. It bound to a lot of different targets and very potent as well at 10 micromolar. CBDA was almost totally inactive. It didn't bind to any targets at all, really. CBCA and CBGA, they were fairly similar to each other. And then lastly, we have CBN, CBDV, and CBC. They clustered together and showed some similarities to each other as well. So, you know, understanding some of these differences in profiles can help us understand potential differences and adverse health effects as well. So this is just done at a single screening. This is a single concentration. So we took out some of the most potent binding at 10 micromolar, and we generate concentration response curves like the one shown here. So here's our IC50 values for those selected cannabinoid target pairs. You can see a lot of them are in the micromolar range, below 10 micromolar. In particular, if you look at DABT, which is dopamine transporter, that had actually the most potent inhibition among all these. That was around 950 nanomolar, and then the other one here is 740 nanomolar. So if you just pick out the dopamine transporter data, look at that a little bit more in detail. Here are the concentration response curves, showing just very typical classic responses here, classic fit. And in case you're wondering, so the dopamine transporter is it's found in the nervous system. And what it does is it pulls dopamine into the nerve cells. So if you block that transporter, dopamine can build up in the nervous system and then kind of saturate all the dopamine receptors. And these are some of the adverse health effects you can expect from that, inhibition. So, among all the cannabinoids, these are probably the most physiologically relevant because they have the lowest IC50s. So this warrants some further study possibly on cell-based assays and looking more at this mechanism. So when we did these concentration response curves, it kind of took us in a little bit new direction, and I'll explain why. So if you look here on the left, these are just typical concentration response curves that you get. And when you fit this data to a Hill, what's called the Hill equation, you have a slope for each one, which describes the shape of the curve. In these cases, the slope is around 1, and this is very typical. However, for a lot of those curves, we found that the slopes were really steep. In these examples, we see how the slope was around 4 and around 8. And a lot of times this indicates there's some sort of issue going on. There's a non-specific effect going on. And in particular, a lot of times this means that there's going to be aggregation occurring. So what's happening, this indicates that the compounds are actually aggregating into colloids, which are causing non-specific effects for target binding. So we wanted to look at this a little bit more in depth. So we used 2 different methods to look at aggregation of these cannabinoids. First, we use dynamic light scattering. It's a physical chemical method. So here we have our cannabinoid concentration in this example increasing, and we can see how the scattering intensity abruptly changes over a certain concentration, indicating that the compound is actually forming these colloidal aggregates. And the point at which there's a huge increase in the scattering is called the critical aggregation concentration. So what we did was we took those cannabinoids and we made solutions in phosphate buffer, tris, PBS, and DMEM, because these are the most common media buffers that are used. And we looked at the scattering of these solutions. And in a second step, we also use an in vitro assay. We took a enzyme inhibition assay for malate dehydrogenase. Historically, this is often used to look at aggregation, which is why we selected it as well. So in this example here, we could see how when we increase the cannabinoid concentration, we see the inhibition increasing as well. However, when we add a detergent, we see that the inhibition basically is abolished, it goes away. So what this indicates is that all this inhibition that we're observing here is due to aggregation, because the detergent, when we're adding detergent, it's actually breaking up those aggregates and it's no longer inhibiting. So from this data here, we can actually use this IC50 from this enzyme inhibition assay as another way to look at aggregation concentration in a more biologically relevant system. So we looked at all the cannabinoids using these 2 different methods, and we found some interesting results. So what we have here are the critical aggregation concentrations here in the various different buffers and media, and here are cannabinoids as columns. So you can see there's quite a difference here in terms of the aggregation concentration. You can see CBC was a very strong aggregator. It aggregated in the lower micromolar range shown here, whereas the acids, CBCA, CBDA, and CBGA, these aggregate at very high concentrations, often above 100 micromolar. So you can see we see a lot of important structural relationships here, where the acid group is really preventing this aggregation from occurring. So if we add in now the malate dehydrogenase inhibition data, which also shows aggregation concentration, we can see similar trends where the CBC is aggregating at the lowest concentration, whereas the acids are fairly high. And finally, aggregation is due to a lot of different reasons, and one of those reasons can be hydrophobicity. And cannabinoids in general are very hydrophobic. So you can see here we have log D values. So these represent hydrophobicity. So higher log D means more hydrophobic. So you can see how CBC is one of the most hydrophobic compounds here, and it also corresponds to the lowest aggregation concentrations. Whereas again, the acidic cannabinoids, they're not as hydrophobic, and they're also aggregating at very high concentrations. So we'd have all this aggregation data. But the important thing to understand is that when we looked at this aggregation, we're using very simple assays. And the conditions in our aggregation assays don't mimic exactly what we do in our biological target screening assays. So these indicate a potential for aggregation in some of those assays, but it's not definitive. You would have to mimic it and reflect it exactly. Because, in a lot of these assays, we have BSA and things, and those can actually affect the aggregation as well. So this is meant to just be a screening tool to, warn or come up with a way to identify potential aggregation in vitro assays. So it's something we have to confirm in the future for individual assays. So this aggregation data, it has some implications beyond this study alone. So what we did was we looked at Chemble, which is this big database containing about 24 bioactivity values, many of which are in vitro. And we searched for the cannabinoids and pulled out the data shown here. So here we have our cannabinoids, different assays. These are just some examples, along with the different bioactivity values expressed as either IC50, EC50, or binding constants. And then what we did was we compared the aggregation concentrations that we observed with these concentrations here for bioactivity. And we found that, for example, CBN, almost half the values in Chemble for CBN are above the aggregation concentrations that we observed. So this indicates potential issues with some of this data because the data from Chemble for these assays could be due to aggregation and not the CBN itself. And again, we don't in these assays for aggregation, we're not mimicking the exact conditions of these assays, so we don't know for sure. But it does kind of open up the question as to whether some of this data could be artifactual. And you can see for CBC and CBDV, we see a lot of data here that also overlaps with aggregation concentrations as well. So finally, I just wanted to mention briefly about some in silico work we did as well. So we have this commercial QSAR model called Chemotargets Clarity, and it predicts binding and concentrations between chemicals, and it can predict binding to almost 5,000 different targets. So we put the cannabinoids through this QSAR model and found some interesting targets that may be of interest for future work. You can see here some examples of these targets we identified. For example, retinoic acid receptors. These are involved in developmental effects. In the last one, this lipoxygenase, this is involved in inflammation. So, these cannabinoids in general are kind of understudied, and these give some ideas for future work. So in conclusion, there's well over 100 cannabinoids out there, and many of these lack safety data. Screening the interactions between cannabinoids and safety-related targets, it's a rather rapid and inexpensive way to identify potential adverse health effects. And despite the similarities among cannabinoids, subtle differences in their structures can drastically change their biological target binding profiles. And dependent on the targets and the concentrations that are being tested, cannabinoids may non-specifically inhibit targets due to aggregation, which could lead to false positives. However, aggregation has to be evaluated using the specific assay conditions and confirmed for each assay. And in general, overall, biological target binding data, it can prioritize subsequent testing, it can support read-across predictions, because it provides important mechanistic data. It can also potentially identify cannabinoids that could contribute to additive adverse health effects through shared mechanisms and targets. So that's the end of my talk today. I just wanted to acknowledge some of the management here at FDA's Human Foods Program. Also acknowledge the scientific computing team for hosting the ChemoTargets Clarity platform. And also wanted to acknowledge collaborators at the University of California, San Francisco, who specialize in aggregation testing. So thank you very much for your attention, and I can entertain questions if we have time.
Yeah, we definitely do. Thank you, Dr. Santillo. We have one question so far. Michael, how did you explore alternative in silico approaches, such as docking, and how robust are the predictions from Clarity? Are things like the training data set and performance statistics available?
Yeah, that's a great question. So first answer is no, we didn't do any docking studies, although they would be useful in some cases. Number two, in terms of the ChemoTargets Clarity, so there's like 5,000 different targets in this program. Some of each one's basically a separate QSAR model, so the predictivity can vary quite a bit. Um, in my opinion, I think the ChemoTargets Clarity is better for kind of initial screening, not really obtaining very exact binding predictions. I think it's a good starting point just to kind of hone in on potentially important targets, but um, I'm not sure if a lot of those targets may be totally accurate. It's something you would have to follow up with. And again, some targets are going to be, some of those target predictions are going to be better than others, depending on how much training data went into it. And within the program, they do show the training data. But with 8,000, with 5,000 different targets, it's hard to go through them all. So if you want to follow up later, I can give you more information to whoever was interested in that.
Okay, great. We have a couple questions on the QSARs now. You sort of touched on this, but they're specifically asking about follow-up verification of the QSAR predictions, such as finding the KI or RAR. I don't know if those.
Yeah, so they're asking about, yeah, so we didn't really go, this is kind of a separate study here. We didn't really do too much follow-up work on these different targets. It was just meant to be an initial screening, um, especially for the retinoic acid receptor, which is RAR. We didn't really look afterwards. We didn't do any in vitro testing of that, but it's something that others might be interested in doing in the future.
Okay, thanks. Is the QSAR prediction of cannabinoids also available for TRPV1?
So yes, it's capable of of predicting TRP1. I have that data, and if you're interested, you can look at um, this article below that contains more details on which cannabinoids were predicted and which ones weren't. So,
Okay, great. And a follow-up question is, can a speaker also comment on how the presented approach can help us understand the mechanism of cannabis hyperemesis, where the causative agents are unclear?
um, I'm not too familiar with that, so I can't really answer. I'm not too familiar with that mechanism
We'll,
or anything.
we'll save it for the discussion, see if
Okay,
anyone has any ideas.
sure.
Um, just another specific question. Do you have a threshold for the IC50 to consider a compound positive or negative? For example, 100 micromolar.
It's hard to develop a really specific threshold. I would mean, and so in general, because we don't know a lot about the ADME properties of some of these cannabinoids. So you can use like relative ranges. So I mean, I would say anything below 1 micromolar could represent more of a risk. We talk more about risks really than really, hard thresholds because I think for a lot of these cannabinoids, the plasma concentrations might be in the tens of, or maybe 100 nanomolar range. So using that as a guide, we can look at some of these interactions as being more physiologically relevant than others. Now, in our screening, we use 10 micromolar as a threshold for doing follow-up IC50s.
Okay.
But that's kind of a separate issue. That was more for prioritizing the experiments, not so much like what's more risky or what's more likely to occur in vivo.
Right. Last question. Does cannabinoid aggregation have any relevance for in vivo bioactivity?
I would say so. In the stump, for example, the cannabinoids, for them to be absorbed solubilized, in the stomach and things like that. Their physical chemical properties are going to play a big role. So I think, that's true with a lot of chemicals, not just with cannabinoids. So that's something that would be, better addressed through maybe like pharmacokinetic modeling and dissolution testing and things like that, looking at how the physical chemical properties can affect the solubility and absorption.
Right. Okay, thanks very much. We're going to
All right.
save the rest of the questions for the group discussion. So then we will move on to the next presenter, Xiugong Gao. Thank you, Michael.
So good morning, everyone. So sorry about the delay. And first, I would like to thank ASCCT for hosting this webinar, and thank you all for joining us today. So I'm going to present you a study we carried out using a human in vitro hepatotoxicity model to derive transcriptomic point of departure for hemp extract and for cannabinoids. So these are some background information that you have already heard of from Yitong's talk. So I will not repeat them here, but just to point out the four cannabinoid we worked on, CBD, CBC, CBG, and CBN. And as you can see from this analysis report, apart from THC, these are the four most abundant cannabinoids in hemp extract. So in this study, we used purified form of these cannabinoids together with hemp extract. So far, the FDA has only approved CBD for the treatment of epilepsy under the trademark Epidiolex. It was reported that during clinical trials for Epidiolex, some patients experienced elevated liver enzymes. And in some of them, the AST level increased so high, they exceeding the 3 times the upper limit of the normal range. So they had to be withdrawn from the trial. So therefore, it was suspected at that time that CBD may cause liver toxicity. So at the beginning of our study, which was around 2021, apart from the clinical trials, there were only less than a dozen published studies on CBD hepatotoxicity. So the 8 animal studies in four species, rats, mice, dogs, and monkeys, consistently showed liver hypertrophy and increased liver enzyme. And the 4 human studies in healthy individuals also showed increased liver enzymes, especially ALT and AST. However, a point of departure could not be established from these available studies. As this report by EFSA concluded, studies are necessary both in humans and in experimental animals that enable the identification of a point of departure. So these are just some refreshers. So in simple words, a point of departure is the transition point between a safe low dose and a dose that starts to cause adverse effect. When we conduct a traditional dose response study in animals, we usually obtain a NOAEL, a non-observed adverse effect level, and a LOEL, lowest observed adverse effect level. So the PAD is usually the NOAEL, and if the NOAEL is not available, we use LOEL for the PAD. However, this approach has some drawbacks, such as being dramatically impacted by experiment design and not accounting for experimental variability. So to overcome these problems, recently a new approach called BMD-BMC, benchmark dose or benchmark concentration, are used to derive PAD. So to do that, we first fit the experiment to a model and then use a predefined change in response called BMR, benchmark rate, to derive the BMD or BMC. And the statistical lower confidence limit called BMDL or BMCL is used as the PAD. So once we obtain the PAD, we can then divide the PAD by a bunch of uncertain factors to obtain the so-called reference dose, RFD, or acceptable daily intake, ADI. Just to point out specifically, the second uncertain factor, UF2, is for animal to human extrapolation. So this is how we derive PAD from apical endpoints of traditional animal studies. We first conduct a subchronic or chronic animal studies, then perform histological or clinical chemistry assays and analyze the data to obtain either a BMDL or a NOAEL as the apical PAD. So the drawbacks of this approach is that they need long durations, usually several months. And also they use a large number of animals. And the final result needs interspecies extrapolation to get the RFD or ADI. So to overcome these shortcomings, there has been a growing trend in recent years to use transcriptomic PAD or TPAD for risk assessment. As outlined in this article published in Toxine in 2022, there are four fundamental principles to use TPAD in risk assessment. First, transcriptomics is a reliable tool for detecting gene expression changes. Second, alterations in gene expression are indicators of biological responses to stressors. Third, transcriptomics can establish a PAD from short-term in vivo studies. And lastly, the use of TPAD will provide a more protective risk assessment. So the TPAD approach was initially designed for short-term animal studies. like a 5-day animal study. So after the exposure, instead of apical endpoint, we measure gene expression in different tissues. And then we conduct a BMD analysis for all the dose response genes. And the lowest gene set, BMDL, is derived and set as the TPAD. So this approach significantly reduces experimental duration and the number of animals used. But by nature, it is still an animal study, and therefore still need interspecies extrapolation. So to be fully NAM compatible, we can use human cell-based in vitro models and similarly conduct a transcriptomic BMD analysis and obtain a gene set, lowest gene set BMDL as a TPAD. So in this case, since we use human cells, so that we don't need to we don't need interspecies extrapolation, but we need in vitro to in vivo extrapolation to translate the TPAD to RFD or ABI. So the objectives of this study is to use toxicogenomics approach combined with a human in vitro hepatotoxin model to study the liver toxicity of hemp extract and for major constituting cannabinoids, and to derive their TPAD values and also to explore their mechanism of action. So this is the in vitro model that we use in this study. Is the ICL hepatocyte 2.0 from Fuji Cellular Dynamics. We will not go into detail, but just to summarize that we characterize the model in 2 transcriptomic studies, and we were able to show that these cells are very similar to hepIg, and in tissue classification, we showed that these cells were more than 80% similar to primary human hepatocyte. So using this model, we first conducted a cytotoxicity study in the concentration range from 10 to 60 micromolar. And we calculate the IC50, and based on this value, we obtain the cytotoxicity potency ranking of the compound in the order of CBN, CBD, CBC, and CBG. And we also calculate IC10 and use these numbers as guide to decide the highest concentration used in transcriptomic study. Basically, the highest concentration was set close to the IC10 value. So for CBN, CBD, and hemp extract, the highest concentration was 20 micromolar. And for CBC and CBG, 40 micromolar was used. And then after zero dilution, a total of 7 to 8 concentration was used in the dose response study. So these are the 3-dimensional PCA plus. In other cases, you can see a concentration responsive effect. So we use the software BMD Express to analyze the data, and this is the workflow. So first, the raw data was loaded into the software, and then the data went through quality control to remove outlier samples, if any. And then the data were normalized to reduce technical variations. Next, the data were pre-filtered to only retain those responsive genes. And then in step 5, all the genes that pass pre-filtering were fitted to 8 different mathematical models. And the best model was selected, and its BMD was selected as the gene-specific BMD. There was also a post-filtering step. This step is we'll remove all polyphotic genes and those genes that having an invalid BMD value. And then in step 8, genes that passed post-filtering were grouped into gene sets, like Go terms. And then the median BMD of all genes in the gene set were taken as the gene set BMD. And finally, the lowest gene set BMD was selected. And its corresponding BMDL was taken as the TPAR. So this is the summary of the modeling result. On the left is the BMC accumulation plot. In general, the steeper the curve, the slope of the curve, the more potent the compound is. So here we have a potency ranking of CBN, CBD, CBC, and CBG. And on the right is the median overall BMC for each compound. And the lower the value, the more potent the compound. So here we have a potency order of CBN, CBD, CBG, and CBC. So different from here, where CBG is the least potent. Here you can see that the CBG is almost as potent as CBD. So the two main reasons for this discrepancy. First, if you look at the the corner of this you can see that for CBG at the lower concentration range, it has a very sharp slope here. And secondly, for comparing potency by median overall BMC, the highest concentration for all the compounds should be the same. But here we use different concentrations for the compounds. So therefore, we devised a formula, try to more accurately evaluated transcriptomic potency. So it accounts for the total number of modeled genes, the highest concentration used, and the median BMC obtained. And using this formula, we obtain the transcriptomic potency of the compound. We set CBD as the base for comparison with an arbitrary value of 1. And you can see that the CBN was more than fivefold as potent than CBD. But CBC and CBG were only about 34 and 14% as potent. So this transcriptomic potency order is in agreement with the cytotoxicity order. So these are the T-part values for the compound together with the associated good terms. But since T-parts are pathway agnostic, so we will not be concerned with the identity of these good terms. But a question that you may ask is how reliable are these values? So we don't have the answer, but a simple way is to compare them with published data. So far, we can only find some data in the literature for CBD. So first, from this publication by Drs. Liu and Sprender from our division, a maximum tissue concentration of 17 different cannabinoids were predicted using PBPK modeling. For CBD, a Cmax of 86.5 nanomolar was reached in liver. Following oral administration of 0.4 milligram per kg, so based on this value, we calculate that the T-part value of 0.106 micromolar for CBD corresponding to an ADI of 0.49 milligram per kg body weight. All right, so this is another publication in 2023, which summarized all available ethical endpoints of clinical and animal studies for CBD. And they reached an ADI of 0.43 milligram per kg body weight. So you can see that these two values are highly concordant, suggesting that the values that we derived are good approximation of ethical part values and are reasonable to use for risk assessment of this cannabinoid. So this slide is just for your information. We are not trying to compare this to our result. So EFSA, the European Food Safety Authority, proposed a new ADI of 2 milligram per day for an average adult of 70 kilogram, which corresponding to 0.0275 milligram per kg, which is very low compared to the value that we obtained. So this value is obtained from BMD modeling of 5 GLP compliant subchronic studies in rat. One reason for the low value they reached is that they applied a very high incidence factor of 400. If you are interested, you can find more information from this paper. So we also perform functional comparison across the 4 cannabinoids. Shown here are a list of canonical pathways, nuclear receptors, disease and disasters, and top tox functions impacted by the 4 cannabinoids. So to summarize this result, each compound was found to impact a unique list with distinctive activation inhibition pattern. And we found that all the 4 cannabinoids were predicted to affect metabolism and to have some beneficial effect on cardiovascular disease, but adverse effect on neural system. And similar to CBD, CBC and CBN display the liver toxicity and the potential to cause cancer, while CBG protected from this adverse effect. We would like to point out that these results were solely based on transcriptomic predictions. So more studies are needed to confirm this result and to fully understand the mechanism of action for these cannabinoids. And I think we are running out of time. I will not explain in detail, but just to let you know that we also compared microarray and iron-seq for the use of deriving TPAD, and we were able to show that these 2 platforms were performed equally well, and they reached TPAD value very similar for both CBC and CBN. So just to summarize, so we use the transcriptomics combined with the in vitro model to derive PAD for the hemp extract and for cannabinoids. And we obtain a potency order for them, CBN, CBD, CBC, CBG, which was consistent with the cytotoxicity order. And we were able to show that the TPAD value for CBD was concordant with the previous published data. And then we showed that each cannabinoid acted through a unique combination of biological processes and pathways with distinctive activation inhibition patterns. But further studies are needed to fully understand the toxicity of this compound. And lastly, uh, we think that the current work demonstrates the potential utility of transcriptomic BMC analysis as a NAM for hazard assessment of dead pore chemicals. So lastly, I would like to thank all my colleagues listed here for their contribution to the project. And I thank you all for the attention, and I'll be happy to answer questions.
Great. Thank you very much, Dr. Gao, for the presentation. We have a few questions here for you. One of them is, did you use the standard iPSC culture medium or a low serum medium for the cytotoxicity test? And they're adding that CBD might bind to serum components present in the medium.
I think we use the standard medium, and we actually analyzed the available concentrations of the cannabinoids in the cell culture, and we were able to show that the binding to protein was low.
Okay,
Yeah.
thank you. Did you determine the in vitro intrinsic clearance of all of the compounds tested in the hepatocyte model? And they're wondering if that could have an impact on the point of departure.
Can you repeat the question?
Yeah, did you determine the in vitro intrinsic clearance of the compounds? And we're wondering if that has an impact on the point of departure.
I'm not quite familiar with the term intrinsic clearance. I don't. I'm not sure what that means. So, but I would say that the concentration we used is the free cannabinoids in the cell culture was close to the nominal concentration that we used.
Okay, thank you. And then I know you presented a slide on functional endpoints, but I think this is different, a little bit of a different meaning of the word functional. So let me ask you the question. In addition to transcriptomic validation of the iPSC-derived hepatocytes, did you look at the functional endpoints such as CYP enzyme activity or albumin production?
I think yeah, this is a good question. I think there are a lot of published people out there trying to characterize this model. So the company itself has a lot of data on that. So we did not characterize the model in terms of function. I mean, we did some studies on that, but our focus here was transcriptomics. Yeah.
That makes sense. Okay, thank you again for your presentation. And we will go to the next presenter. So
I'm
we have
going
Dr.
to.
Kirsten Eckstrum.
Hi, can you hear me and see my slides?
Yep, everything looks good.
Awesome. Okay. So I'm Kirsten Eckstrum. I'm a research biologist in the Division of Toxicology. And today I'm going to be talking about our work examining the hepatotoxic potential of cannabidiol or CBD, cannabidiol containing hemp extract, and cannabinol or CBN at consumer relevant concentrations in primary human hepatocytes. So all of the work that I'm going to be presenting today is actually published in the Journal of Applied Toxicology. And here's the reference if you would like to look that up either during the presentation or after or anything. So there are a lot of cannabinoid and hemp-derived products on the market, and some of these products have hemp extract in them. And there are 120 known phytocannabinoids in hemp extract. The most abundant and well-studied of these is cannabidiol or CBD, and the first isolated is cannabinol. And these are two of the compounds that we're going to be looking at in more detail, along with the hemp extract. Other than phytocannabinoids, there's also other constituents in hemp extract that can potentially influence the toxicity of the products, such as terpenes and toxic elements. So recently a survey was conducted on US hemp-derived products, and it showed that there's actually a pretty large range of concentrations of cannabinoids in these products. The lowest was 0 milligrams per serving. So essentially products are saying they have cannabinoids in them, but there's none detectable. The highest was 143 milligrams per serving, and the median was about 16 milligrams per serving. And this is across a wide range of products, including gummies, beverages, pet products, edibles, oils, and powders. And interestingly, fewer than half of these products that were surveyed contained CBD concentrations that were within 20% of what was on their label. And as I mentioned, CBD is the most well-studied of the cannabinoids, and this is partially due to its research in the clinical setting. As other people have mentioned, Epidiolex is an FDA-approved cannabidiol medication that's used in the treatment of seizure disorders. And it's typically administered in concentrations of 1 to 20 milligrams per kilogram per day. And at these concentrations, the maximum concentration of CBD observed in the bloodstream after administration, or the Cmax, is about 1 to 3 micromolar. And at these concentrations, there is the potential for hepatotoxicity, and this is observed with an increase in liver enzymes such as alanine transaminase, or ALT, and aspartate aminotransferase, or AST. And this is indicative of liver injury in the clinical setting. Hepatotoxicity is also seen more frequently when the CBD is co-administered with another seizure medication called valproate. So this is showing that in the clinical setting, there has been observed hepatotoxicity with CBD exposure. But what about with healthy individuals? So another clinical study was performed in which 1,500 milligrams per day was administered to healthy adults, which equates to about the 20 milligram per kilogram per day for a 70 kilogram weight adult. And in this study, they also saw increased liver enzymes. 31% of the participants had elevated levels at 5 times the upper limit of normal, which is consistent with drug-induced liver injury. However, there was large interparticipant variation where some of the participants had this DILI effect and others did not. So in clinical settings, we see that there's the potential for hepatotoxicity due to CBD exposure. But what about in vitro systems? So over the next couple of slides, I'm going to talk about some of the in vitro studies that have been done with cannabinoids. And in this study, one of the ways that we can look at hepatotoxicity in vitro is with the use of a human liver cell line called HepG2. So in this study, they exposed these cells to CBD and CBDV, and they saw a dose-dependent decrease in viability at 50 micromolar for CBD and 162 for CBDV. And at subcytotoxic levels, they were also able to see an increase in DNA damage from 2 to 18 micromolar after 24 hours. So this is showing that, one, we can see hepatotoxicity with CBD in culture, and that there can be some changes at at lower concentrations than effects on viability with higher concentrations. Another cell line that is often used for detecting hepatotoxicity are human liver HepaRG cells. And these cells can be formed into spheroids, which was what was done in this study. And when these HepaRG spheroids were exposed to CBD, they calculated the IC50 using ATP at 24 hours to be about 86 micromolar, and at 72 hours to be about 58 micromolar. They also did a transcript analysis and demonstrated that there was an increase in the number of altered genes at around 10 micromolar, showing that, again, there's some changes that can be happening prior to cytotoxicity in these models. Next, this study compared the HepG2 cells to primary human hepatocytes, which we're looking at in our study. And primary human hepatocytes are cells that are collected from human liver donors. So here we can see that there's a dose-dependent decrease in ATP in both the primary human hepatocytes and the HepG2 cells, but the effect is stronger in the HepG2 cells. This study also did a benchmark concentration analysis and determined that the BMC50 was about 33 micromolar for the HepG2 cells, whereas it was about 54 micromolar for the primary human hepatocytes. They also saw that there was an increase in cytotoxicity, which is measured with lactate dehydrogenase, or LDH, in HepG2 cells. And the final study I want to talk about is a more complex system. So this study used a quad liver chip microphysiological system, which consists of primary human hepatocytes, liver sinusoidal endothelial cells, Kupffer cells, and stellate cells. And they exposed their chips to CBD and CBN at lower concentrations of 0.24 for the low dose, 3 and 4.7 micromolar for the high dose over a 7-day period. And they saw some transient effects with albumin and LDH, so there's some variability over time and between experiments. So these data are really demonstrating that there are some potential changes that can be observed in culture with exposure to cannabinoids. However, the potential hepatotoxicity due to consumer relevant exposure to hemp and its ingredients is relatively unknown. So that is our goal to answer that question. So we split our study into 2 main parts. First is to determine the consumer relevant concentrations that we should be exposing ourselves to and determine a chemical analysis of the extracts. And then second, we are going to be exposing these cells to primary human hepatocytes and exposing the primary human hepatocytes to the chemicals and evaluating the hepatotoxicity. So, So first we did in silico modeling to determine the in vitro concentrations, and this work was done by Yitong. And she selected a 30 milligram serving size of CBD by oral exposure to calculate these values. And 30 milligrams was chosen because it is consistent with the amount of CBD in products on the market. So in order to do this calculation, she predicted the concentration and time curves of CBD in plasma and other tissues. So this is this figure here. Plasma is the red line shown here, and the liver is actually the blue line. So calculating the Cmaxes, the plasma Cmax was 6 nanomolar, and the liver Cmax was about 86 nanomolar. So this is really showing that the liver is seeing a little bit more of the cannabinoid exposure. And we can use this liver Cmax value to help us determine what concentration our hepatocytes should be seeing in vitro. For comparison, since we're also looking at CBN, 30 milligram exposure to CBN would result in a plasma Cmax of 9.1 nanomolar and a liver Cmax of 129.2 nanomolar. So again, the liver is seeing a higher concentration of the drug. So we use the in silico modeling to help us determine the dose range. And for all of our compounds, CBD, CBN, and the hemp extract, we're actually going to keep all of the doses the same so that everything is directly comparable. And we're using the CBD for in silico modeling to help determine those concentrations. So at the low end, we're exposing ourselves to 10 nanomolar, and at the high end to 25 micromolar. And this is so that we are encompassing the predicted liver Cmax from the 30 milligram exposure to CBD. And we're also getting a little bit of that clinical exposure to CBD. Epidiolex, again, led to a 1 to 3 micromolar clinical plasma concentration, or the Cmax, plasma Cmax of 1 to 3 micromolar. And that would equate to about a 14 to 43 micromolar concentration for the liver. So it might seem a little odd that we are stopping at 25 micromolar instead of going by a factor of 10, as it seems to be the trend. But at concentrations higher than 25 micromolar, the CBD and the hemp extract was actually falling out of the media, so we had to lower the concentration. So next we did a chemical analysis of the hemp extract. So the hemp extract that we received was an ethanolic hemp extract from the University of Mississippi. And because ethanol can have some effects on the liver, we evaporated out the ethanol and reconstituted in DMSO. And shown in this table are the concentrations of the 5 highest constituents in the hemp extract. So first is CBD, then CBC, CBG, CBDV, and last CBN. And you can see that the concentration of CBD is much higher than the rest of the constituents. So now that we determined what the constituents in our extract were, we wanted to make sure that the chemicals and the constituents were stable in our cell culture media. So in order to do this, we prepared our media and the concentrations in the media, and then immediately extracted out the chemicals for LC-MS detection. So in these tables, you can see the concentrations that we use. And then on the right-hand side, this is the average percent recovery. And that recovery is the amount of the chemical compared to the predicted concentration or the nominal concentration. So for these higher concentrations, we were able to recover about 70 to 75% of the CBD in the pure CBD extract, meaning we're losing about 25 to 30% of the compound. And at the lower concentrations, we actually didn't detect any of the compound. For pure CBN, similarly to the pure CBD, at the higher concentrations, we were getting about 75% recovery, but the lower concentrations were actually pretty good. And then for the CBD matched hemp extract, CBD recovery was pretty good. For CBN, we have about 70% of the compound being recovered at the higher concentrations, whereas at the lower concentrations, we were not able to detect the chemical. For CBC and CBG, there was actually pretty good recovery of the compounds, except at the concentrations where we were not able to detect the chemical. And for CBDV, we had about 60 to 70% recovery with these concentrations, the higher concentrations, and at the lower concentrations, we were not able to detect the chemical. So some interesting things I want to point out. CBD recovery was much better with the hemp extract compared to the pure CBD. So it could be that there's something in the hemp extract that's helping to stabilize the CBD in our media. And next, I just wanted to remind everyone that the CBN in the hemp extract is not the same concentration as the pure CBN because the CBN is at a lower concentration in the hemp extract, so that those are not directly comparable. So now that we've determined the stability of our compounds in the media, we could do our hepatotoxicity assessment. To do this, we had 4 different donors for 4 different experiments, and we plated our primary human hepatocytes in a 24-well plate. After 6 hours, the cells are covered with a extracellular matrix called Matrigel, which helps the cells to maintain their structure and function. 24 hours after the plating, the media was refreshed, and another 24 hours after that, the cells were treated with their respective chemicals, and then either collected at 24-hour or 48-hour. And there was no retreatment of chemical during any of this time point. So time 0 is the only treatment of the cells. Again, the chemicals that we're using are CBD, CBN, and hemp extract. All of the concentrations are the same. We have 0, 0.01, 0.1, 1, 10, and 25 micromolar of each of the chemicals. And we also included a positive assay control, usnic acid, at 50 micromolar. And this is just to make sure that our assays and our endpoints are functioning as they should be. So the endpoints that we looked at are listed here, and they were evaluated at both 24 and 48 hours. For cytotoxicity markers, we used LDH release, which is a cell membrane permeability marker, caspase 3.7, which marks apoptosis, and then we had functional effect markers, which are basically telling us if our hepatocytes are functioning like they should be. And for that, we have albumin secretion and urea secretion. For mitochondrial markers, we have TMRM, which is a marker of mitochondrial membrane potential. And finally, we evaluated the number of nuclei using NucBlue. So first, we'll talk about the cytotoxicity and apoptosis. And we didn't really see a lot of effects here. So LDH is collected from the supernatant, and with increased hepatotoxicity, we should see an increase in LDH. So in this figure, we actually inverted the data. So an increase in LDH is actually a decrease in viability, which is what is seen with our positive assay controls here. But the cannabinoids largely did not affect viability. Similarly, with the caspase 3.7, an increase in apoptosis would result in an increase in fluorescence, but we inverted the data so that it would actually show up on our graphs as a decrease in viability, but there were no changes with caspase 3.7. Next, we looked at hepatocyte function using albumin and urea, and both of these endpoints are collected from the supernatant, and with hepatotoxicity, they should be decreased, which is what is seen with our positive assay control at all time points and with both albumin and urea. But for our cannabinoids, we saw some small changes. There was reduced albumin with the 25 micromolar concentration of CBD, which is the light gray bar or the first bar here, of about 16%, and a decrease with our hemp extract, which is the dark gray bar or in the middle bar, of about 26%. And then CBN, which is the final bar in the lightest gray, there was no effect. And interestingly, this was seen at 24-hour, but not at 48-hour. Similarly, with the urea, we saw that there was a small reduction in urea at the 25 micromolar concentration at 24 hours of about 10%, and this wasn't carried over to the 48-hour time point. And then CBD and CBN had no effects on urea. So next we looked at mitochondrial membrane potential. So TMRM should be decreased with toxicity, which is what is seen with our positive assay control. And at 24 hours, we saw a decrease in TMRM of the 25 micromolar concentration of hemp extract, no effect on the CBD or CBN. And at 48-hour, we didn't see this decrease in TMRM anymore, but also we saw that there was a little bit of donor variability, especially with the CBD and the CBN endpoints or concentrations, chemicals. And where two of the donors were actually showed a dose-dependent increase in TMRM and two did not. So there was no significance here due to this variability. We also looked at nuke blue and did not see much effect in nuclei quantification. So in conclusion, there was two main parts of our study with some significant results. And first is the chemical stability, and second is our hepatotoxicity assessment. And in the chemical stability, we saw decreased stability of the cannabinoids in the media, which could suggest that the hepatocytes did not receive the full concentration of cannabinoids that were presented to them, which could be why we didn't see as much of an effect in our hepatotoxicity assessments. Also, interestingly, we saw that CBD was more stable in the hemp extract than the CBD alone, again, which could indicate that there's something in the hemp extract that was helping to stabilize the CBD in our media. And also, it could be why we saw some slightly more toxic effects with the extract compared to the CBD alone. For our hepatotoxicity assessment, we didn't see a lot of effects on viability or cytotoxicity, but we did see a couple of effects on hepatocyte function. The 25 micromolar concentration of CBD and hemp extract decreased albumin, and the 25 micromolar concentration of hemp extract decreased urea. And interestingly, this happened at the 24-hour time point, but not at the 48-hour time point, which could indicate that since the cells were treated with the chemicals at time 0, perhaps by 48 hours, the hepatocytes have already metabolized the chemicals and are beginning to recover. We also saw that there wasn't any effects or any toxicity at less than 10 micromolar for any of our endpoints. And we also saw that there is a considerable amount of donor variability, especially with TMRM. Donor variability is something that's pretty common with the use of primary human hepatocytes. But if you recall from some of the clinical studies, there was also variability in participants. So this donor variability could be representative of clinical variability as well. So with that, I'll thank the people who actually did the work. Annalise Stritz was the principal investigator on this project. Kira did a lot of the hepatotoxicity assessments. And then the chemistry team of Yang, Esatir, and Corey did the hemp extract chemical analysis, and Yitong did the in silico modeling. So that's all I've got. If you've got any questions, I'd be happy to answer those.
Yeah, thanks, Dr. Eckstrum. We do have a question so far. Everyone, please feel free to add your questions in the Q&A. And I also want to pull your attention to the answered column. If you open up the Q&A, you will see open answered as 2 columns. And when you hit answered, some of the speakers have been adding in written responses to some of the questions. So you may want to check that out as well. Okay, Dr. Eckstrum, for your questions. The first one is that CBD is known to introduce apoptosis, particularly by increasing caspase 3-7 activity. Is there any reason why no significant difference was observed in primary human hepatocytes?
Yeah, so that's it. Definitely something that kind of surprised us a little bit. I think that there's a number of things that could have happened. It could be that the lower concentrations of the chemical were having an effect, or like they weren't receiving the right amount of the concentration. And primary hepatocytes do seem to be a little bit less sensitive to the cannabinoids, so that could be a part of it as well.
Okay, thank you. Next question is, was any serum used in the culture medium? And if so, did you observe any effects of the serum, particularly on cytotoxicity?
Um, no. So this is actually something that I had to ask the primary investigator about. I don't think that this media contains serum in it. So that there are other ingredients within the media that could be binding with the chemicals, but it seems like the serum was not one of the ingredients that was doing that. We use a serum-free media.
Okay, great. Thank you. Thanks again for your presentation. That's all we have at this point for questions. If there are any more questions that come in, we should have a few minutes at the end to address them. Okay, and with that, we'll go to the last but certainly not least presenter, Dr. Piper Hunt. I can see your slide, so that looks good.
There we go. Can you hear me?
Yes.
Great. So first, I want to thank the organizers, given that this work is on C. elegans. It is neither cellular nor computational, so I'm especially honored to be asked to speak today. And on this study that we did on CBD, as well as the same hemp extract that Kirsten did her studies on. So just briefly, I don't want to spend a lot of time on this, but if anyone wants to ask any questions after about the model, why should anyone care about data from a microscopic worm that has no liver, no skeleton, no eyes? And the reason that you should care is because you can look at development in this model very easily and quickly. They have a 3-day life cycle. So they grow from hatched worm to adult egg-laying worm in 3 days. And because they are clear, you can observe effects on oocyte maturation. Many of the mechanisms of oocyte maturation are conserved from humans to worms. And you can also observe the embryonic development as the fertilized eggs move closer and closer to the vulva for egg laying. And I want to point out that while C. elegans does not have a notochord, many of the processes involved in this invagination are also involved in mammals, indicating that this model can be used to develop to model developmental effects. These are just a small list of pathways that are conserved between C. elegans and humans that are involved in development. Additionally, this fantastic study led by Wendy Boyd and Jonathan Friedman showed that for over 200 chemicals in the ToxCast data set, that C. elegans growth predicted rat and rabbit developmental effects nearly as well as those two models predicted each other. Just briefly, I want to mention that C. elegans is primarily an oral toxicity model, and given that we work for the Human Foods Program, that's a key feature. They have microvilli. They use most of the same digestive enzymes that we do. They have peristalsis. Focusing on the model in general, because of their short lifespan, you can look at chronic and acute toxicity at different life stages. For this project, we developed an emulsification method, which allowed for oral exposure to lipids within a aqueous media system. So in comparison to the other small model organism for NAM methods of zebrafish embryos, while the zebrafish have a circulatory system, a liver, a two-chamber heart, The embryos are an absorption model, whereas, as I mentioned, C. elegans is primarily an oral toxicity model. And while all pathways are not conserved, those controlling morphogenesis, organismal development, neurotransmitter synthesis, function, the neuromuscular junction architecture is highly conserved. And that's all in a in vivo organism. But as I present my data, please remember that it's not a perfect model for humans, especially the uterus. It does not correspond to mammalian uterine function. And like all in vitro testing, there are issues with solubility, volatility, and I'll talk a little bit about adsorption to plastic today. And while people tout that 70% of C. elegans genes have human orthologs, that leaves 30% that do not. I just want to briefly mention PAX6, which controls eye development in mammals. It was originally thought when the C. elegans genome was decoded that there was no PAX6 gene, but a functional PAX6 that had just the portion of the active binding portions has since been found. And so I think as time goes by, we'll find that orthologs, even if they're not homologous along the entire gene, we'll find more of them. I don't want to go too much into how endocannabinoid signaling is conserved. Mike mentioned CB1, and the C. elegans ortholog is NPR19, and the human CB1 can replace the mutant gene and restore behavioral responses to endocannabinoids. And this was a fun study looking at appetitive and consumatory feeding behaviors, which basically they looked at preferred foods in humans and worms. This made the rounds in popular media saying that worms get the munchies too because cannabinoids, exogenous cannabinoids, stimulate increased feeding of preferred foods as opposed to not preferred foods in both humans and worms. When we first started doing this study, we were working with DMSO, and I couldn't figure out why my data was all over the place. I normally get beautiful dose response curves. I wasn't getting them. And our valiant chem team, who we rely on, was able to find that actually the higher the concentration of CBD in C. elegans habitation medium, the more plastic adsorption there was. And so instead, we turned to using emulsion-based dosing exposures, using 280 at a final exposure concentration of 0.1%. Milk is the emulsifier, and milk is a component of C. elegans habitation medium. So it's something they're used to eating as their protein source as they grow. And so using a microfluidizer to produce those emulsions. And so this is my fat-free control. And the 10x dosing solution would have been 1% between 80 in cow's milk, which, as I mentioned, makes up 20% of C. elegans habitation medium. And the effective diameter of the particles was at about 200 nanomolar. C. elegans, right after they hatch, their buccal cavity has a 1 micrometer approximately diameter, and that's the right size for their preferred food in the lab, which is normally E. coli. Our lab does not use E. coli as a food source because E. coli do have their own metabolism. And so by using C. elegans habitation medium, we avoid that. But I use it here to show that the size of these, the worms are actually eating the particles in the emulsion. E. coli also have a slight negative charge, and the particles within our emulsions were approximately in the range of that negative charge of E. coli. So the data on the next two slides that I'm going to show you focuses on C. elegans development. The worms were initiated exposures right after first feeding after hatching, and they grow through 4 larval stages, which are well defined in terms of morphology and behavior, into an egg-laying adult in about 3 days. Exposures were for 24 hours or continuous, and the data I'm going to show you is the timing to the 3rd larval stage relative to controls, as well as the locomotor activity at the 3rd larval stage, which gives a time for post-exposure effects. Before I show you the CBD data, I want to just first quickly show you what an irreversible developmental toxicant looks like at the 3rd larval stage. So here we have 5-fluorouracil, and this so this is the control in side-by-side plates, and it gives you an idea of the variability within the system, which is about 5%. And so that's our minimum for biological relevance. And you can see that there's a dose response that when you remove the 5-fluorouracil, the delays to the 3rd larval stage, and also the data I didn't show here, but to the 4th larval stage were also very similar, whether you took the 5-fluorouracil away or not. Looking instead at a partly reversible toxicant, ribavirin is my positive control for reversibility, and you see that by taking the ribavirin away after 24 hours, that there is a definite reduction across a range of exposures in the delay, although the delays are still there. And so when I did the CBD exposures, once we finally worked out the emulsion exposure system, I was expecting to see one of these 2 modalities. But what I got instead was an increase in delay. And so while we were not looking at levels of endocannabinoids, what I suspect is happening here is that there is a refractory period or a desensitization of endocannabinoid, either synthesis or signaling or receptor molecules, that is then leading to a temporary increased delay after you remove the CBD. We saw a similar effect, although at higher concentrations with hemp, indicating that within this system, hemp extract that was normalized to internal CBD concentration was slightly less toxic than CBD alone. And this was true across all C. elegans assays that were utilized in this study. CBD had a hypoactivity effect in C. elegans that were exposed continuously from hatching. But when you remove the exposure, that hypoactivity was lost. And so again, these were apical effect studies. And so what we can say is that in all likelihood, that the pathways that are affected by CBD that influence locomotion are different than the pathways where CBD is affecting developmental timing. And again, the hemp extract matched for concentration for CBD had less of an effect, whether continuous or for 24 hours on locomotion. So we know that high-fat diets are associated with decreased activity in humans and mice, although causation has not been determined for that. And we also know that many models of human diseases which are associated with hypolocomotion, such as arthritic inflammation, liver disease, and Parkinson's disease, when you expose those models to CBD, that the locomotion parameters are improved. And we saw a similar thing with C. elegans, where here we have our vehicle control emulsion, which is actually a high-fat diet for this model, versus their oil-free matched control. And there was hypoactivity with the emulsions, which the final exposure was 0.5% sesame oil within the emulsion. But when we added CBD, there was actually a recovery at the lower concentrations of normal locomotion parameters, indicating concordance with mammals for this restoration of spontaneous locomotor activity with CBD in adults. So we had hypoactivity with the juveniles, and we had restoration of normal activity with the adults. So looking here at reproduction, we're exposing the worms right at the young adult stage, which is defined as them having a visible primary oocyte, but no fertilized eggs within the uterus. So that's when exposures began. And exposures were for 4 days, with single wells being taken out to be tested at 2 and 3 and 4 days post-exposure. And so everything was normalized to the vehicle control emulsion. And here's our positive control, which inhibits reproduction. Epidural is used as a contraceptive in C. elegans experiments. And what we see is that 400 micrograms per mil reduced the reproductive output with CBD, and that the concentration, as I mentioned, was always slightly higher with hemp extract. And with hemp extract, there may or may not, I wish we had been able to do more experiments with this, there may have been some recovery that was not seen with the CBD alone. So here we're looking at just the progeny. So these experiments were done in a microfluidic instrument designed to accommodate the size of C. elegans. And so again, we're looking at 2 days post-parental exposure at the time that they reach the young adult stage, 3 days and 4 days. And you can see that the longer the exposure, the more you're seeing a reduction in the size of progeny. And so, so here, the progeny ratio is decreasing at 400 micrograms per mil, which is the same concentration that we saw toxicity in the adults. But the effect on the progeny, especially those that were exposed during the time that they were that eggs were being developed, was half that. And again, the concentration was higher with the hemp extract. So high-fat diets cause oxidative stress in humans and mammals. And this strain uses a glutathione S-transferase that's controlled by the C. elegans ortholog of NRF2 called SKIN1. And this model will fluoresce green in the intestine when exposed to oxidative stress, indicating activation of NRF2 regulated genes. And so here I'm using instead the fat-free control, because high-fat diets cause oxidative stress in people. And we see that this vehicle control emulsion does increase oxidative stress threefold in this model. And so here we're looking at transgene activation of expression and time of flight, which is an indication of the C. elegans size. So there was no difference in size between the oil-free and the high-fat diet control. And the concentrations that reduced oxidative stress were much lower than the concentrations that induced toxicity. Again, in the adults, we saw toxicity at 400 micrograms per mil with CBD. There was reduced toxicity with the hemp extract. So here, we wanted to compare the concentration of CBD in C. elegans with something. It doesn't really work to compare it with serum concentrations in humans. And of course, we don't have a whole lot of data about tissue deposition from humans. So we're focusing on mammalian data instead. This is a 28-day rat study. And for comparison, Epidiolex, the recommended exposures are 5 to 25 mg per kg per day. And this child and Talon study was looking at about that, the top concentration, about 5 times that and about 8 times that. And what they found was that at these higher concentrations for CBD exposures, there was about 100 milligrams per kg of CBD in adipose tissue. And C. elegans have fat storage molecules, fat storage organelles in their intestine, which which can be corresponded to lipid storage in mammals. And so we found, of course, if you're looking at CBD per exposed worms, these were all 24-hour exposures. So these worms were exposed from the time they hatched, and then the adults were exposed for 24 hours prior to evaluation. Using estimated values of worm density and volume, we found that at 300 micrograms per mil for 24-hour exposures, the tissue concentrations were about 2 to 4 times this study that looked at 5 times the recommended exposure of Epidiolex. And so on the one hand, you could say that this is an indication of the safety of CBD in this model. On the other hand, Yitong talked about how CBD is being sold in gummy bears, where each one often has an adult dose. And so there's rising incidence of calls to poison control centers and emergency room visits where children are eating a whole package of those candies. And so I think that this data shows that while these exposures are very high, the difference between the juvenile responses to the adult responses indicates that what's safe for adults for CBD is not necessarily safe for children. So just in summary, look, if we take oxidative stress response as an indication of efficacy, those concentrations were in the 25 to 100 micrograms per mil range, which were 8-fold lower than the toxic concentrations in adults. That the adult toxic concentrations were twice that for juveniles. CBD induced hypoactivity in juveniles, but it normalized locomotion in adults. And so again, that's a life stage difference. There was similar uptake levels of CBD that was purified versus from hemp extract, but across all of our studies, there was a slightly less toxicity with hemp extract, and that actually corresponds to, there've only been a couple of studies that have actually compared one-to-one purified CBD versus hemp extract in humans. And those studies did find that there were fewer adverse events, gastrointestinal adverse events with hemp extract relative to matched concentrations of purified CBD. So the main findings from this study that I feel are most important is that there was an effect that would indicate that there was a refractory period or a downregulation of endocannabinoid signaling systems after removal of CBD. And importantly, these toxic findings are below human recommended dosing, but potentially not below toxic exposures from candy-like products. And so I'll stop there and take questions.
Thank you, Dr. Hunt. I don't see any questions coming in yet for you, but I'm sure one will pop up soon. While we're waiting for that, maybe we can come back to some of the other questions that we have in our. Oh, here we go. Okay. Sorry, Dr. Hunt, we do have a question for you. I knew if I waited just a couple minutes, it would come in. What stage in human development does L3 delay in C. elegans correspond to?
That is a fantastic question. There is a huge expansion of cells and cellular specificity during the third larval stage. And so it could be considered analogous to a prepubertal state. So they're not reproductively competent in the third larval stage, but there's this massive restructuring of the gonad that goes from a very small portion of the worm to taking up about a quarter of the volume of the worm. And so in my mind, it's a pre-pubertal stage.
Okay. Thank you.
But I want to stress that while they have endocrine signaling, tissue to tissue signaling, they don't have an endocrine system per se. They don't have gonadotropin hormones, and so saying that it's a prepubertal stage is with a big caveat that they're not mammals.
Right. Thank you. There's another question. Can you say anything about the metabolic capacity of C. elegans?
That is also a great question. They have lots of cytochrome C enzymes. They share many digestive enzymes. Kirsten worked with me on this previously, where we were looking at homologs of human liver enzymes in C. elegans, which I still hope we can publish one day. Thank you, Kirsten, for working on that with me. And so on the one hand, yes. On the other hand, is that metabolism the same as humans? That depends on the chemical. Sometimes it is, sometimes it isn't. And so while there is a huge amount of data showing that it is, when you look at chemicals such as cyclophosphamide, which is actually a pre-drug, and it needs to be activated within the human liver to become a chemotherapeutic, that is negative in C. elegans. And so I would be careful. I would want to know compound by compound. If I was looking for specific metabolites, I would want to look at those individually before I said for sure.
Okay. And the next question is, do we know, or do you know, I guess, if the findings have been reported in other animals? Some of the findings.
I'm really excited about this, and I hope that as time goes by that will be reported. There are some other beautiful studies in C. elegans with cannabinoids, but they were all in adults. And so this study was the first to report this this developmental effect of removing CBD and having increased delays. And I think that will, because endocannabinoids endocannabinoid signaling is so important in human embryonic development, also postnatal development, also pubertal development. I would love to see that, but it hasn't happened yet, as far as I know.
Okay, thank you. So we have a few minutes left. Thank you for your presentation again. We have a few minutes left where we can address maybe some questions for the entire group. And I have saved one question, which I'll get started with and see if more come in. So again, if you have a remaining question, maybe about the session as a whole, can we have the whole group go ahead and turn on your video? We'll have maybe a couple minute panel discussion. So this question is, and I will confess, I'm not really sure if any of you are going to be able to answer it, but I'm going to ask it anyway. And how does the recent rescheduling of THC into Category 3 in the US affect the specific NRMs used in testing for these adverse effects? Does anyone have any thoughts about that? Looks like no. That's okay. It's a little bit outside of your specialties. I will also wanted to just turn everyone's attention to a couple of the discussions that have been going on in the answered questions. And there was one question about that we didn't quite have a discussion about. When the question was, it would be interesting to compare points of departure obtained in primary rat hepatocytes to NOELs obtained in 90-day studies in rats. And the answer was just to clarify that Dr. Guo's presentation was using human cells. But yes, that could be an interesting comparison. I don't know if anyone else wanted to say anything more about that. I think for me, when I think about this question, actually, we've had experience with trying to compare in vitro and in vivo, even in the same species. Sometimes it doesn't quite work. Of course, you have to build in vitro to in vivo extrapolation, but you also have to think about the fact that the dosing and the kind of experimental design and a lot of other factors are different. So, with those caveats, it could be a way to help gain confidence in vitro assays, but it also could be misconstrued, if we're not considering all of the details. Sorry, Dr. Gao, go ahead. I see you have your mic off.
Yeah, I think that is true. So the question is trying to see if in vitro and in vivo study result concordant with each other. So that being said, I think the result from our study shows that these two systems are quite concordant. Although these are quite different systems, but bear in mind that the point of departure, as long as they are within an order of magnitude, I mean, by a factor of 10, we consider them to be concordant.
Okay, thank you. We've got another question coming in, and I'll leave this open to anyone who wants to address it. How confident are you in using NAMs for risk in a regulatory environment?
Can I start really quick?
Right.
I'm glad that you said risk, uh, because NAMs are great for detecting hazard. And one of the key things that the Division of Toxicology is at the Human Foods Program is trying to work on is how can we get to predicting safety? Hazard and risk, I think that NAMs are great at that. I like the comparison to the Ames test because the Ames test, in looking at the sensitivity and specificity of that test, if it's positive, you're pretty sure that that compound is going to break DNA or do something to harm the DNA, right? But if the Ames test is negative, it doesn't tell you that it's not a carcinogen. And so I feel like that's where we are right now. The in vitro testing and the small alternative models, we know a lot. If it's positive, we're pretty sure. But if it's negative, there's less assurance that that's going to be negative in humans.
I think, did anyone else want to add to that?
I would like to add a little bit. So, at FDA, in 2024, FDA did publish a roadmap trying to kind of, reduce animal testing in clinical studies and then to encourage NAM submission. So at least in HFP, the Human Foods Program, and CEDAR, we do have NAMs qualification programs. And then any sponsors would like to qualify their NAMs for regulatory use can submit their materials to FDA. So to kind of help FDA to understand more about their NAMs method and then how it has to be fit for purpose. It's not like a general like a platform. We qualify the platform for any use in foods and drugs. It's more like for a very specific purpose. Is this for predicting metabolites or anything else? And it has to be fit for purpose. So we do have the program. And then if you're interested, you can check out FDA's website. It's called iStand and Entree. I stands for CEDAR and Entree for HFP, if you're interested.
Great. Okay, well, thanks to you both. I think that's all the time we have. Again, I want to really appreciate Dr. Liu for putting this session together. I'm really glad we were able to have it, even despite last fall's government shutdown. And so thank you all for tuning in and giving your talks. And thanks to all of you listening as well and asking great questions. And we will see you at our next webinar. So be sure to go to our website to find that out. And as we said, yes, these slides and the recording will be available on the web as well. Thank you all. Take care. Bye-bye.
Thank you very much.
Thank you very
Bye.
much. Bye-bye.